Hi everyone, welcome back to Logical Pharmacology. By the end of this video, you'll be able to recall every sympathetic response and receptor type without cramming. Let's get straight to it. Picture Jack walking down the street. A dog suddenly jumps out and barks. Jack panics, and his sympathetic nervous system kicks into full alarm. Classic fight or flight. Norepinephrine and epinephrine flood the postganglionic nerve endings, and tissues across the body are either stimulated or inhibited, depending on whether they help Jack survive. Here's the general rule of thumb. Tissues that are useful under stress get stimulated. Muscles contract, sphincters constrict and contract, vessels constrict, neuroendocrine glands are activated and increase secretion. These are the words you want to remember when you think of responses of effector organs that are helpful. Tissues that aren't immediately helpful are inhibited, muscles relax, vessels dilate, glands cut back on secretion, and so on. Now, to the receptors themselves. The organs that are stimulated in this helpful way. Here I have colored green for helpful. These mostly have alpha-1 receptors, with a few exceptions. The heart, the juxtaglomerular apparatus in the kidney, and pineal gland. These three work mainly through beta-1 receptors. Another exception is the brown adipose tissue, which expresses beta-3 receptors predominantly. Older text sometimes lumps this under beta-1-like action, but physiologically, beta-3 is the correct receptor. On the flip side, the not-so-helpful organs that are inhibited are generally under beta-2 receptors, except for three special cases: autonomic nerve endings, platelets, and some parts of GIT. They all have alpha-2 receptors. And then we have pancreas. I don't say it is an exception. I say it is a beautiful coincidence. In fact, think of everything opposite of what the general rule is. Alpha cells of pancreas are stimulated by beta-2 receptors, and beta cells of pancreas are inhibited by alpha-2 receptors. These receptors, alpha and beta, are what we call adrenergic receptors or adrenoceptors. They're proteins sitting on the surface of cells, and their job is to respond to epinephrine and norepinephrine. We split adrenergic receptors into two big families, alpha and beta. Each of those is then divided further: alpha-1, alpha-2, beta-1, beta-2, and beta-3. And even within alpha-1, there are smaller subtypes like alpha-1A, alpha-1B, and alpha-1D. And within alpha-2, there are subtypes alpha-2A, alpha-2B, alpha-2C. All right, let's jump back into Jack's story and see what's going on, organ by organ. This story literally lets you remember every single function of the autonomic nervous system. First up, those tiny Arrector Pili muscles. When the postganglionic neurons release norepinephrine, they contract, pulling the little hairs on the skin upright. That's what we call goose flesh or piloerection. In animals, this makes them look bigger. In humans, it's more about thermoregulation and emotional responses, about the receptor. Since the muscles are contracting here, they've got alpha-1 receptors. Now Jack needs his pupils wide open. That is possible when the radial pupillary dilator muscles contract, pulling the edges of the pupil outward, called mydriasis or pupil dilation. Contraction, so alpha-1 receptors. Next, the lens. Normally the human lens is biconvex, but it is not static, it is dynamic. Normally the ciliary muscles are contracted, the suspensory ligaments are relaxed, and the lens is biconvex, or rather globular in shape, to be able to see nearby objects clearly. The lens are attached to the ciliary muscles through suspensory ligaments. In a stress response, however, Jack wants far vision, to see all his options for escape. Here's how it works. The ciliary muscles relax, the suspensory ligaments are pulled tight, and the lens goes flat. That's what's needed for far vision. Relaxation means the ciliary muscles use beta-2 receptors. And while we're still at the eye, Arousal also lifts the eyelids. Sympathetic activation contracts Mueller's muscle, that little accessory smooth muscle in the upper eyelid. This pulls the lid up and widens the eye, giving Jack that wide-eyed, alert look. Again, contraction here is stimulation. So that's alpha-1 receptors. Next, the salivary glands. Under sympathetic stimulation, they don't pump out the usual watery saliva you'd get from parasympathetic activity. Instead, they produce a smaller amount of thick, viscous saliva packed with proteins and enzymes. That makes sense in fight or flight. Jack is not sitting down to enjoy a meal. He is prepping the mouth with protective proteins and amylase to quickly lubricate and break down anything if needed. This shift in composition also explains why your mouth feels dry when you're stressed. It's not a lack of saliva entirely, but the fact that it's thicker and there's less of it. Stimulation, as the general rule, means alpha-1 receptors. Now here's an interesting one. The pineal gland doesn't actually ramp up the sympathetic response. In fact, it helps tone it down a little bit. Here's how it works. Norepinephrine released from sympathetic nerve fibers stimulates the pineal gland to produce melatonin. That melatonin then travels to the hypothalamus and boosts GABAergic inhibition in the paraventricular nucleus, PVN. This actually dampens sympathetic vasomotor tone, a sympatholytic effect. That means better blood pressure regulation and a protective buffer against excessive sympathetic activity, like during orthostatic stress. Although basically pineal gland is being stimulated to synthesize melatonin, it uses the beta receptor. The first exception we've come across for organs stimulated by alpha-1 receptors. Sympathetic outflow to the skin is split into two main types of postganglionic neurons. Cholinergic neurons, which innervate the sweat glands. These cause increased sweat secretion. That's stimulation, so they're linked with alpha-1 receptors. Sweat secretion here tends to be more localized, like on the palms. That's why we call it adrenergic sweating. Now you might be thinking, hang on, why adrenergic if the neurons actually use acetylcholine? Here's the trick. During development, sweat glands start off with norepinephrine. This norepinephrine acts on adrenergic receptors in the sweat gland, triggering a developmental change. The sweat gland then pushes the neurons to switch to a cholinergic phenotype, so they begin releasing acetylcholine instead. At the same time, adrenergic neurons innervate skin blood vessels, causing vasoconstriction through alpha-1 receptors. That's why the skin looks pale under stress. Blood gets redirected to organs that matter more in a fight or flight scenario. Adrenergic neurons also activate the Arrector Pili muscles, making those little hairs stand upright using the alpha-1 receptors. Next, for Jack to run, he needs more oxygen in, more carbon dioxide out, so his airways need to open right up. That means less secretion and less interstitial fluid, less swelling of the mucosa and submucosa, less hyperemia, relaxed bronchi and trachea, constricted bronchial blood vessels. This is a tricky one because the constriction of the bronchial vessels redirects blood away to organs like the heart, skeletal muscles, and brain. This ensures that blood is being redirected to more vital organs, and in fact, oxygen-rich blood is now being sent more efficiently. Here, the minor blood supply to the airways is temporarily being compromised for the greater systemic benefit. Okay, so the bronchial blood vessels constrict. That's stimulation via alpha-1 receptors. The mast cell membranes get stabilized, reducing histamine release and inflammation. That's inhibition via beta-2 receptors. And finally, the tracheal and bronchial smooth muscles relax to widen the airways. Blood gets redistributed. Skin and GIT vessels constrict through alpha-1. Renal blood flow drops. Jack isn't worried about peeing mid-escape. Again via alpha-1. Abdominal viscera follow the same pattern. Skeletal muscles are the opposite. They initially constrict but circulating epinephrine activates beta-2 receptors, causing vasodilation. Local metabolites like nitric oxide and adenosine enhance this, a phenomenon called functional sympatholysis, making sure muscles get maximum blood flow. Coronary and cerebral vessels are mostly unchanged, regulated by local metabolic autoregulation. When the sympathetic nervous system kicks in during stress, say when blood pressure drops, it directly stimulates the kidneys, specifically the juxtaglomerular cells, to release renin into the bloodstream. Renin then sets off the renin-angiotensin-aldosterone system, or RAAS, in the bloodstream by converting angiotensinogen into angiotensin 1. Next, angiotensin-converting enzyme, or ACE, steps in and turns angiotensin 1 into angiotensin 2. Angiotensin 2 is a powerful vasoconstrictor. It tightens blood vessels, boosts systemic vascular resistance, and raises blood pressure. It also stimulates the release of aldosterone and antidiuretic hormone, ADH, both of which tell the kidneys to hold on to more sodium and water, increasing blood volume. And to top it off, angiotensin 2 feeds back to the central nervous system, ramping up sympathetic outflow even further, creating a reinforcing loop that stabilizes blood pressure under stress. So here the juxtaglomerular cells are stimulated. Stimulation should be via alpha-1, but since it is an exception to the rule, we know it has beta-1 receptors. The heart, on the other hand, fires right up. This leads to positive inotropic, positive chronotropic, positive dromotropic, and positive bathmotropic effect. Heart is being stimulated here, but it mainly runs on beta-1 receptor exception number 3. These effects are seen all over, including the SA node, AV node, atria, ventricles, and his Purkinje fibers. Overall, the heart works harder and faster. About 80% of cardiac adrenoceptors are beta-1, 20% are beta-2, and a few alpha-1 for protective functions in heart failure. Digestion slows right down. Sympathetic activation reduces absorptive and secretory activity in the stomach because Jack's body needs that energy elsewhere. Pyloric and ileocecal sphincters contract, that is, alpha-1 receptors. Longitudinal muscle layer relaxes, which means beta-2 receptors. Secretions, including the HCl, pepsinogen, and gastrin, are inhibited via alpha-2 receptors. Remember, the GIT is one of those exceptions where inhibition comes from alpha-2 rather than beta-2. Again, digestion is not a priority, so the wall of the gallbladder relaxes through activation of beta-2 receptors, causing bile to be stored and concentrated. The sphincter of Oddi, on the other hand, contracts through alpha-1 receptors. This increases resistance, preventing bile from flowing into the duodenum and diverting it to the gallbladder. This slows bile release into the intestine. The spleen contracts, squeezing out stored blood and platelets into circulation. This boosts oxygen delivery and enhances clotting, handy in case of injury. The contraction is mediated by alpha-1 receptors. Pancreas. Sympathetic stimulation excites the alpha cells in the islets through beta-2 receptors. This causes an increase in the release of glucagon. Glucagon drives glycogen breakdown and gluconeogenesis in the liver, raising blood glucose. At the same time, sympathetic nerves inhibit the beta cells via alpha-2 receptors, suppressing insulin release, so glucose stays in the blood for immediate use by muscles and the brain. And sympathetic activity also causes vasoconstriction through alpha receptors in the pancreas. Upon this, the pancreatic acini, the clustered pancreatic cells responsible for releasing digestive enzymes into the small intestine, experience an overall inhibition. When the sympathetic nervous system ramps up, the posterior pituitary gets involved too. However, it does so indirectly, not through adrenergic receptors. The posterior pituitary, triggered by hypothalamic input, releases vasopressin and oxytocin. Vasopressin constricts vessels and increases water reabsorption, maintaining blood pressure and volume during stress. That way, vital organs like the brain and muscles keep getting enough blood supply and are able to maintain perfusion during stress or hemorrhage. Platelets actually have a bit of a side role in the sympathetic story. They mainly carry alpha-2 adrenergic receptors, with just a few beta receptors. When epinephrine hits these alpha-2 receptors, the platelets get activated and start clumping together. That's part of the normal hemostatic and clotting response. But here's where it gets interesting. Once activated, platelets release serotonin, and this can influence nerve activity in ways that don't exactly support the sympathetic fight or flight mode. For example, during carotid plaque formation, serotonin released from platelets can trigger a reflex that actually inhibits sympathetic activity and lowers blood pressure, kind of the opposite of what the sympathetic system is trying to do. It's thought to be a feedback mechanism to keep things in check. Under stress, the body wants you to hold your urine, though in bladder dysfunction, some people may leak. The internal ureteral sphincter, responsible for closing the bladder neck and preventing urine leakage, contracts via alpha-1 receptors. The detrusor muscles, on the other hand, relax via beta-2 receptors. This combination holds urine in. The ureters also respond. They're rich in alpha-1 receptors, especially in the distal part near the bladder. When stimulated, intracellular calcium rises, making the smooth muscle contract harder. This increases peristalsis, helping urine move more forcefully toward the bladder. Sometimes the contractions are so strong they can become spasms, like during a kidney stone. The genital system responds in a way that it always promotes sexual activity and protection of the product of conception, that is, the fetus. In males, sympathetic postganglionics stimulate the prostate and seminal vesicles via alpha-1 receptors to release their secretions, the ejaculation. But remember, this only happens after an erection, which is a parasympathetic job. In females, sympathetic stimulation causes rhythmic contractions of the vagina and uterus. In a non-pregnant woman, this is linked to sexual arousal and orgasm. Naturally, the contractions are meant to help with semen transport. These contractions are driven by alpha-1 receptors on uterine smooth muscle. In pregnancy, it's different. High estrogen and progesterone levels suppress alpha-1 receptor expression, make the beta-2 more dominant in myometrium of the uterus. Beta-2 activity prevents contractions, keeping the uterus relaxed. That's why historically, beta-2 agonists like Ritodrine and isoxsuprine were used to suppress preterm labor, although they are no longer recommended as first-line tocolytics due to their side effects. Muscles normally store glucose as glycogen. During stress, glycogen storage is inhibited via beta-2 receptors, so glucose stays free for energy. Now, constant contractions during escape, like Jack sprinting from the dog, require rapid ion shifts. With every depolarization, sodium rushes in. With every repolarization, potassium moves out. This would quickly deplete potassium if not corrected. Here, sympathetic stimulation boosts the sodium-potassium ATPase pumps, restoring ion balance. And here's another neat exception. This contractility boost and ATPase activity in skeletal muscle is mediated by beta-2 receptors, not alpha-1. The sympathetic system also fuels the body by mobilizing energy. Glucose. Epinephrine and cortisol stimulate the liver and muscle cells to break down glycogen into glucose, glycogenolysis, and to make new glucose, gluconeogenesis. Blood glucose rises sharply, giving muscles and brain immediate fuel. Similarly, lactate is produced during glycolysis in the skeletal muscles and can be sent to tissues like the heart, brain, and slow-twitch muscle fibers for use. During stress or exercise, the brain may even prefer lactate to glucose. I want to make this very clear. Logically thinking these two, The glucose and lactate are a product of skeletal muscles contraction, right? So the receptor involved here should be similar to the skeletal muscles, that is the beta 2 receptor. In fact, I could have mentioned this when mentioning the skeletal muscles. Adrenaline released from the postganglionic fibers in adipose tissue stimulates the breakdown of triglycerides into free fatty acids and glycerol, a process called lipolysis. This is one of the very important exceptions. Happens via beta 3 receptors found in adipose tissues. The fatty acids supply long-lasting fuel, while glycerol goes to the liver as a glucose precursor. That wraps up our discussion on adrenergic receptors and the sympathetic responses they drive. In the next video, we'll switch gears to the cholinergic receptors and the parasympathetic system. So stick around, because I promise you'll enjoy the step-by-step, no cramming approach as we logify pharmacology together.